Hello and good morning, everyone. My name is SHARLINA binti AHMAD SHARIZAL AZUAR, matrix number 1891824, and I will be your chairperson for today. Thank you for coming. Today, we will be discussing about enzyme kinetics. But before I proceed further, let us all introduce ourselves, starting from my right.
My name is Tan Wei Yang and my metric number is 188722.
Hello everyone, my name is Tan Iwan, matrix number 189724.
Hi everyone, my name is SYAZWANI binti LOKMAN. My matrix number 190197.
Hi, my name is ATIFA binti Muhammad APANDI and my metric number is 189744.
Hello, my name is NURUL AQILAH binti Muhammad SOIB and my metric number is 190507.
Hello everyone, my name is NADHIRAH binti ABDUL HAMID and my matrix number is 186861.
Hey, my name is ALISSA SYAMIRA binti JAHABAHALI and my matrix number is 190274.
Hi, good evening. My name is AQILAH binti ANNUAR and my metric number is 187312.
Hi, my name is FARHANAH binti ZAINAL ABIDIN, matrix number 1901.
Okay, so now that the introductions are done, we will proceed. Would anyone like to go first?
I would like to start our discussion to explain about what is actually an enzyme and its function and also examples. So enzymes is basically are multi-protein complexes made up of a number of individual protein subunits, and they have this molecular weight which is the sum of the atomic weight of a molecule's atom, which is ranging from about 10,000 to more than 1 million. And some enzymes have this molecular weight that fall below the range listed above. So the main function of the enzyme is actually to catalyze the reaction that occur in our body or animals. So in other words, they speed up the reaction by providing an alternative reaction pathway of lower activation energy. And we know that the enzyme has this specificity where the enzyme reacts specifically with only one substrate, which they will react at the active site of the enzyme, which is substrate will bind towards the enzyme to reduce the activation energy. And this enzyme can be classified according to the reaction involved and end with ACE. For example, maltase substrate is the maltase is the substrate for the maltose, or hydrolysis is the enzyme for the hydrolysis reaction. So we can know easily by looking at the spelling of the word, which is when they end with ACE, so we can know that it is an enzyme. And these enzymes are actually proteins. Like the protein, the enzyme contains chains of amino acid linked together, and the characteristic of an enzyme is determined by the sequence of amino acid arrangement. When the bonds between the amino acids are weak, they may be broken by the condition of high temperature or high level of acids. So when these bonds are broken, the enzyme might become non-functional. So the enzyme take part in the chemical reaction do not undergo permanent changes, and hence they remain unchanged to an end of the reaction.
So now I will be talking about enzyme function. So enzymes are biological molecules. They are typically proteins that significantly speed up the rate of the chemical reactions that take place within the cells. So enzymes are important for the body function, such as aiding in digestion and metabolism. So some enzymes help break large molecules into smaller pieces that are more easily absorbed by the body, and other enzymes help bind two molecules together to produce a new molecule. Enzymes are highly selective catalysts, meaning that each enzyme only speeds up a specific reaction. The molecules that an enzyme works with are called substrates. The substrates bind to a region on the enzyme called the active site. There are two theories explain the enzyme substrate interaction, which are the lock and key model and induced fit model. So in the lock and key model, the active site of the enzyme is precisely shaped to hold specific substrates. In the induced fit model, the active site and substrate do not fit perfectly together. Instead, they both alter their shape to connect. Whether the case duration that occur accelerate greatly over a millionfold once the substrate bind to the active site of the enzyme, the chemical reaction result in a new product or molecule that then separates from the enzyme, which goes on to catalyze other reactions. Here are the example. When the salivary enzyme amylase bind to a starch, it catalyzes hydrolysis, the breakdown of a compound due to the reaction with water, resulting in maltose or malt sugars. So that's all for the enzyme function.
Now I will talk about why the structure of the enzyme is so important to its function. Most enzymes are proteins and therefore their function is specific to their structure. Enzymes function as a catalyst to increase the rate of virtually all the chemical reactions that take place in a living system. The enzymes, like all catalysts, are not consumed but are constantly used to catalyze the same specific reaction. Enzymes depend on the correct structural alignment and orientation at the active site of the protein and the appropriate site of the reactants or substrate before the reaction can be proceeded. This geometric interaction between the enzyme and the substrates is referred to as the lock and key model because the enzyme's action parallels the action of a lock, into which is fitted the key or substrate. If the key and lock do not match, the action does not work. It is the same with enzymes and substrates. The active site for the enzyme and the appropriately matched site of the substrate must physically join before the reaction can occur. That is why the structure of the enzyme is so important to its function. The enzyme binds with the appropriate substrate only in the correct alignment and orientation to connect the molecules. The resulting enzyme substrate complex enables the reaction to occur. Finally, the products are formed and the enzyme is released to catalyze the same reaction for another substrate of the same type of molecule. Enzymes may fail to function if they are denatured. Remember, the model simplifies your understanding of the process. In reality, they are three-dimensional molecules.
Alright, I'll be explaining about what is enzyme kinetic. Enzyme kinetic is the study of chemical reactions that are catalyzed by enzymes. In enzyme kinetics, the reaction rate is measured and the effects of varying the condition of the reaction are investigated. Studying an enzyme's kinetic in this way can reveal the catalytic mechanism of this enzyme, its role in metabolism, how its activity and control, and how a drug or an agonist may inhibit the enzyme. Enzymes are usually protein molecules that manipulate other molecules that enzymes substrates. These target molecules bind to an enzyme's active site and are transformed into products through a series of steps known as the enzymatic mechanism. This mechanism can be divided into single substrate and multiple substrate mechanism. Kinetic studies on enzymes that only bind one substrate, such as trisulfosphate isomerase, aim to measure the affinity with the enzyme binds this substrate and the turnover rate. Some other examples of enzymes are phosphorylfructokinase and hexokinase, both of which are important for the cellular respiration glycolysis. When enzymes bind multiple substrates such as dihydrogenase reductase, enzyme kinetics can also show the sequence in which these substrates bind and the sequence in which products are released. An example of an enzyme that bind a single substrate and release multiple products are proteases, which cleave one protein substrate into two polypeptide products. Other joins two substrates together, such as DNA polymerase linking a nucleotide to DNA. Although these mechanisms are often a complex series of steps, there is a typical one rate determining step that determines the overall kinetics. This rate determining step may be a chemical reaction or a conformation change of the enzyme or substrate, such as those involved in the release of products from the enzyme.
Okay, so I will be continuing the discussion by talking about activation energy. So by definition, activation energy is the least amount of energy required to activate atoms or molecules to a state in which they can undergo a chemical reaction. So basically, activation energy is the minimum amount of energy to activate those atoms or molecules to condition in which they can undergo chemical transformation or physical transports, basically involved in all reactions in the biochemical terms and field and such. So in transition state theory, the activation energy is the difference in energy content between atoms or molecules in activated or transition state configuration and the corresponding atoms and molecules in the initial configuration. So activation energy is usually represented by symbol E subscript A in mathematical expressions, usually to measure the reaction rate constant. So activation energy of a chemical reaction is kind of like that hump you have to get over to get yourself out of bed. So even energy releasing or exergonic reactions require some amount of energy input to get going before they can proceed with their energy releasing steps. So, okay. So enzymes in cells speed up chemical reactions by lowering the activation energy that is needed to start the reaction. So imagine activation energy as the hump on top of a hill. Pushing a rock over the hump requires energy. So once the rock is over the hump, energy is released as the rock rolls to the bottom of the hill. So in relation to enzymes, activation energy is lowered with the help of enzymes. I believe will be explained later by other people in the discussion. So yeah, that's all for me. Thank you.
Actually, I would like to continue what ALISSA talked about in terms of how enzymes actually lower activation energy. So we all know in order for a reaction to occur, activation energy, which is the minimum energy requirements needed, have to be met. And this can be done by effective collisions. So before I actually move on to how enzymes do it, I will just talk about the first 5 factors that actually affect the rate of reaction. So first of all is the concentration of the substrate. This is because when the number of molecules per square area increases, they will hit each other more. So the number of collisions actually increase. And when these collisions increase, the number of effective collisions will increase. And this will increase the rate of reaction. Next, we have the smaller the particle is, the smaller the total surface area is, which means more area is exposed. And this leads to higher collisions and higher effective collisions, leading to higher rate of reaction. Next, we have high temperature. The higher the temperature, the higher the kinetic energy of the particles. So they will actually hit each other more and also hit the sides of the object itself or the container. And it will actually cause more collisions, which again increases high effective collisions, and it will increase the rate of reaction. Then we also have high pressure, although this actually just applies for gas. So what happens is when the pressure is high, the gases are compressed and they will be actually hitting each other more, the gas particles, and they will also be hitting the walls of the container, which actually again increases high collision, high effective collision, and increases rate of reaction. You can increase the pressure by either reducing the size of the object itself or increasing the temperature, but this only applies for gas again. Now I will specifically focus on the last part of how how to increase the rate of reaction, which is catalysts, in our case enzymes, which actually lower activation energy. How they do this is that they actually position the substrates and adjust the bonds to become unstable and reactive. So you don't need that much activation energy because all the bonds are already unstable. And that is how they lower the activation energy bond. So in our case, again, to recall, the 5 factors that affect the rate of reaction is concentration, total surface area, temperature, pressure, and catalyst, which in our case are enzymes.
So I will be talking about the enzyme assays. So what is enzyme assays? Enzyme assays are performed to serve two different purposes, which is to identify a special enzyme to prove its presence or absence in a distinct specimen like an organism or a tissue, and second is to determine the amount of the enzyme in the sample. While for the first, the qualitative approach, a clear positive or negative result is sufficient. The second, the quantitative approach must deliver data as exact as possible. A great advantage of enzyme is that they can be identified by their catalyzed reaction, in contrast to the other components of the cell, like functional proteins or nucleic acid, which must be determined by direct detection. During the enzyme reaction, product accumulates in amount exceeding by far the intrinsic enzyme concentration. However, the conclusion from the product form back to the amount of enzyme in the sample comprises various difficulty and pitfalls. There are 4 types. For enzyme assays, the quantity or concentration of enzyme can be expressed in molar amounts. And there are 4 types of enzyme assay, which is first is initial rate experiments, and then progress curve experiments, transient kinetic experiments, and relaxation experiments. So that's all from me. Thank you.
Sorry to interrupt, but I would just like to know, how can you tell the rate of reaction from a graph? Like, do you calculate the gradient, or do you watch how it curves? Or yeah, that's all I know.
I think SAFWA can answer your question, Sharlina. SAFWA, why don't you explain on how to determine the rate of reaction from the graph?
So I'll be talking about the rate of rate in the presence and absence of enzymes, and these experiments have been done in our lab, and the experiment runs at about 22 degrees Celsius and using a smart spectrometer for quantitative analysis. Because since the enzyme is very highly temperature dependent, and changes in room temperature can have an effect on the rate of reaction relative to the results. Increasing in mind, room temperature will actually increase the reaction rate and can lead to saturation of adsorbent at an earlier reaction time point. Conversely, a colder room also will cause the temperature to slower the reaction rate. So in front of me is the whiteboard that contains that I have written about the data collected during our experiment that we have done before. So the amount of here is the quantitative and qualitative data that has been collected. There isn't much difference from the data collected, and the curve also show about same reading. However, at the 4 minute until to 6 minutes for the qualitative data, the results show more rapid increase, and then at the 6 minute and 8 minute it just gives a constant reading, bit different than the qualitative, a bit different from the quantitative data that increase uniformly. And about the since why we are doing this thing actually is the standard curve of the product p-neutrophenol is related to the amount of p-neutrophenol present in a cubic, and by plotting the absorbance values for standard with known amounts of p-neutrophenol, it is to determine how much p-neutrophenol is present in our enzyme assay sample. And by comparing the standard curve with the value for the enzyme, like what we did from the data, make it more possible to match the absorbance with the amount of p-neutrophenol. So, at the end of the experiment, we can actually see the initial rate of product formation with enzyme present for quality and quantity terms are showing not much difference. The quality data shows about 12.5 nanomole per minute, meanwhile the quantity shows.
Thank you, SAFWA. So I will continue my explanation on enzyme assay, which is the two sampling method of enzyme assay, which is a continuous assay where the assay give a continuous reading of activity, and this continuous assay where samples are taken, the reaction stop, and then the concentration of substrate or product determined. For continuous assay, it is the most convenient with one assay giving the rate of reaction with no further work necessary. There are many different types of the continuous assay, which is spectrophotometric, fluorometric, colorimetric, chemiluminescent, light scattering, and microscale thermophoresis. Meanwhile, for discontinuous assay, it is usually taken from an enzyme reaction at intervals, and the amount of product production or substrate consumption is measured in the sample, which is radiometric and chromatographic.
So for this experiment, we have to calculate the rate of the enzymatic reactions. So in this experiment, we did it in 3 different temperatures, which are 0 degrees Celsius and 37 degrees Celsius and 60 degrees Celsius. So we already plot the graph by using the reading we got in the experiment. So we have the graph function already, and we have the equations. So now we already have the y, the number of the y already. So we want to determine the x by using the equation that we got from the graph. So let's take an examples. So when we want to find the x at the temperatures of 0 degrees Celsius, so we put the y value, which is 0.148 inside there, and then we will get that x is equal to 6.929. So same goes with 37 degrees Celsius and 60 degrees Celsius. So the x will be 23.267 and 22.506. Okay, after we get the x reading, so now we can calculate the rate of reaction for that particular temperature. So for 0 degrees Celsius, so we use the x value, deduct by the initial value, which is 0 nanomole, and then we divide by the total times, which is 2 minutes, and the initial time definitely is 0, right? So we divide by that, and we will get 3.465 nanomole per minute, and that is the rate of reaction for that particular temperature. So generally that is the way to calculate the rate of reactions for this experiment. So thank you.
So now I will talk about an industry where enzyme kinetics is applied. Since enzymes are powerful biochemical catalysts, they are widely used in the food industry and more in manufacturing other chemicals. For instance, enzymes in yeast cells convert sugar into ethanol and carbon dioxide in the brewing and drinks industry. A similar process is used to convert sugarcane into ethanol, and later it will be distilled to be used as biofuel. In the industry, enzymes are used to bring about reactions at normal temperatures and pressures that would otherwise require more expensive and more energy demanding equipment. Some of the examples of enzyme usage is enzyme proteases, which breaks down proteins and are used to pre-digest the protein in some baby foods, while enzyme invertase is used to make sugar for soft chocolate. Other than that, pectinase breaks down insoluble pectin polysaccharides and so is used to clarify fruit juice. Enzymes are also used in genetic engineering and penicillin production. Last but not least, the dairy industry uses enzymes made by good bacteria to produce yogurt and cheese from milk. The bacteria enzymes convert the sugar in milk, which is lactose, into lactic acid. Thank you.
Okay, I believe we have come to the end of our discussion. Thank you so much to everyone for contributing, and I hope this discussion will help you in your future as vets in training. And I think that is all we can conclude about enzyme kinetics today. Thank you all for coming, and have a good day.